Welcome to the vast, quiet expanse of Starcom. Here, we look at the wonders of our world through a cosmic lens, finding the extraordinary in the familiar. Tonight, we turn our gaze toward a creature that has shared our hearts while keeping its soul in the shadows of the wild, the cat. Beyond the soft fur and the gentle purr lies a biological enigma, a predator refined by millions of years of evolution to be the ultimate observer. In this 2-hour immersion, we explore 100 dimensions of the feline existence. From the quantum mechanics of their movement to the celestial myths that bound them to the stars in ancient times, this is a journey into the heart of a mystery. As the world slows down around you, tell us in the comments, from which corner of our planet are you drifting with us tonight? What hour of the night is it for you? If you seek clarity in the calm, join the Starcom community by subscribing. The feline pupil, a gateway to nocturnal mastery. The vertical slit pupil of the domestic cat represents one of evolution's most elegant solutions to predatory vision. Unlike circular pupils found in diurnal hunters, the cat's elliptical aperture can dilate to occupy approximately 85% of the visible eye surface in darkness, while contracting to a mere thread in bright sunlight. This remarkable dynamic range, spanning a factor of nearly 300, allows cats to hunt effectively across lighting conditions that would render most predators helpless. The vertical orientation creates an asymmetric depth of field, keeping both near and far objects in simultaneous focus along the horizontal plane, precisely the geometry needed to judge the distance to prey moving across the ground. Recent computational models have revealed that this pupil shape also reduces chromatic aberration more effectively than circular apertures, sharpening the image where it matters most. The slit's ability to close so completely protects the sensitive retina from damage during daylight hours, when the cat's raw-dense retina would otherwise be overwhelmed. This pupillary architecture appears predominantly in ambush predators of small stature, suggesting a deep connection between hunting strategy, body size, and ocular evolution across the carnivore family tree. The tapetum lucidum, nature's biological mirror. Beneath the feline retina lies a crystalline layer called the tapetum lucidum, a structure so optically sophisticated it resembles engineered nanotechnology. Composed of 15 to 20 layers of zinc-cysteine complexes arranged in microscopic ribbed patterns, this biological mirror reflects light back through the photoreceptor cells, giving them a second opportunity to capture photons. The result is a 6-fold improvement in low-light vision compared to humans, allowing cats to hunt effectively in conditions we would perceive as near total darkness. The tapetum's interference patterns also produce the characteristic eyeshine observed when light strikes a cat's eyes at night, a glow that has haunted human imagination for millennia. The color of this eyeshine varies by individual, ranging from gold to green to blue, determined by the precise mineral composition and structural spacing of the tapetum layers. Interestingly, Siamese cats often lack a tapetum lucidum entirely, a genetic peculiarity linked to their distinctive albinism genes. The evolutionary investment in this structure speaks to the primacy of crepuscular hunting in feline ancestry, where success during dawn and dusk demanded every possible photonic advantage over alert, keen-eyed prey species. Rod-dominated retinal architecture. The feline retina contains approximately 25 rods for every cone photoreceptor, a ratio 6 times higher than that found in human eyes. This rod-dominated architecture sacrifices color discrimination and detail resolution in bright light for extraordinary sensitivity to motion and contrast in dim environments. Cats possess only 2 types of cone cells, making them functional dichromats similar to humans with red-green color blindness. They perceive the world primarily in blues, greens, and grays, with reds and pinks appearing as variations of gray or muddy brown. However, this apparent limitation belies a sophisticated visual system optimized for detecting movement. The high density of rods makes feline vision exquisitely sensitive to flicker and motion, allowing cats to detect prey movements that would be invisible to human observers. Each rod cell connects to a dedicated ganglion cell through a direct neural pathway, creating a rapid signal transmission system that operates with minimal processing delay. This neurological architecture enables reaction times approaching the physical limits of nerve conduction velocity. The evolutionary trade-off is clear. Cats exchanged the chromatically rich, detail-oriented vision of diurnal primates for a motion detection and low-light system unmatched among terrestrial predators. Whisker proprioception and spatial navigation. The mystacial vibrissae, commonly called whiskers, constitute a sensory array of extraordinary sophistication, each follicle embedded in a blood sinus surrounded by hundreds of mechanoreceptor neurons. These specialized hairs, called tylotric follicles, respond to deflections as minute as 2 micrometers, allowing cats to detect air current changes around obstacles in complete darkness. The whisker array maps precisely to the somatosensory cortex in a topographic pattern called the barrel cortex, where each whisker has a dedicated column of neurons processing its input. This neural architecture enables cats to construct three-dimensional spatial maps of their immediate environment without visual input, essentially seeing through touch at a distance. The whiskers extend approximately to the width of the cat's body, providing a constant gauge for whether openings are navigable. During hunting, cats protract their whiskers forward, creating a tactile cage around captured prey to monitor its movements and position the killing bite with precision. Recent studies have demonstrated that cats can discriminate between objects differing by less than 1 millimeter using whisker sensation alone, a tactile acuity rivaling human fingertip sensitivity. The whisker system operates with such efficiency that it consumes significant cortical real estate, reflecting its evolutionary importance to feline survival. The Jacobson's organ and chemical communication. Housed in the roof of the feline mouth lies the vomeronasal organ, also called Jacobson's organ, a chemosensory structure dedicated to detecting pheromones and other non-volatile chemical compounds. When a cat performs the flamen response, curling back the lips and opening the mouth in an apparent grimace, it's drawing air across a pair of small ducts behind the upper incisors that connect to this specialized sensory apparatus. Unlike the olfactory epithelium that processes volatile odors, the vomeronasal organ analyzes heavier molecules that convey reproductive status, territorial claims, and individual identity markers. The sensory neurons within this organ project directly to the accessory olfactory bulb and then to the amygdala and hypothalamus, bypassing the cortical processing that conscious smell requires. This direct connection to emotional and reproductive centers enables pheromone signals to trigger immediate behavioral responses without conscious interpretation. Male cats exhibit the flamen response most frequently when investigating female urine, but both sexes use this sense to read the complex chemical messages left by other cats. The persistence of this ancient chemosensory system in modern cats reflects the continued importance of scent communication in feline social structure, even in domestic environments where visual and auditory signals might seem sufficient. Purring, the multifaceted vibration. The mechanism by which cats produce their characteristic purr remained mysterious until the late 20th century, when high-speed imaging revealed the process involves rapid twitching of the laryngeal muscles at frequencies between 25 and 50 Hz. These contractions open and close the glottis approximately 25 times per second, creating pressure oscillations in the airflow during both inhalation and exhalation. This continuous, phase-independent sound production is unique among vocalizations, requiring no breaks for breathing. The frequency range of purring coincides precisely with vibration frequencies shown to promote bone density and accelerate healing of soft tissue injuries in mammalian systems. Research has demonstrated that exposure to mechanical vibrations between 20 and 50 Hz stimulates osteoblast activity and enhances fracture healing, leading some researchers to hypothesize that purring evolved as a self-healing mechanism for an animal that spends substantial time inactive. Cats purr not only in contentment but also when injured, frightened, or dying, supporting this therapeutic interpretation. The energy cost of purring is remarkably low, making it an efficient healing modality. Some evidence suggests that the vibrations may also benefit humans, with studies indicating that cat owners show reduced risk of cardiovascular disease and lower stress hormone levels, possibly mediated by exposure to these therapeutic frequencies. Feline jaw architecture and the carnassial shear. The skull of the domestic cat showcases a mandibular structure optimized for delivering a killing bite with minimal force application. The temporomandibular joint sits nearly level with the tooth row, creating a mechanical advantage where jaw muscles work with maximum efficiency. Unlike omnivores with grinding molars, cats possess highly specialized carnassial teeth, the upper fourth premolar and lower first molar, that function as biological scissors, sliding past each other with a shearing action that slices through flesh and tendon. This dental adaptation eliminates the need for chewing, allowing cats to process food through cutting alone. The canine teeth, long and slightly recurved, are designed for precision rather than crushing power. Cats possess an extraordinary proprioceptive sense in these teeth, with sensory receptors that allow them to feel the spaces between cervical vertebrae when delivering a killing bite to prey. This explains the remarkable consistency with which cats strike the precise location needed to sever the spinal cord. The reduced number of teeth compared to ancestral carnivores, only 30 in adults versus 42 in dogs, reflects a continued evolutionary refinement towards specialized killing rather than generalist feeding. The clavicle reduction and arboreal agility. Among the many skeletal adaptations that enable feline agility, the vestigial clavicle stands out as a masterpiece of evolutionary compromise. Unlike humans with their firmly connected collarbones, cats possess only a small, free-floating bone embedded in muscle tissue where the clavicle would normally attach to the shoulder. This reduction in skeletal connection between the forelimbs and axial skeleton allows the shoulder blades to move independently and dramatically, permitting a stride length that exceeds what rigid skeletal connections would allow. During a run, the cat's shoulders can rotate through a range exceeding 60 degrees, effectively lengthening each stride and allowing the cat to place its rear feet in the exact footprints left by the front feet. This narrow gait pattern minimizes sound and visible tracks while maximizing efficiency. The lack of a rigid clavicle also allows cats to compress their chest and shoulders when squeezing through tight spaces, making them capable of navigating any opening through which they can fit their head. This adaptation proves particularly advantageous for arboreal locomotion, where varying angles and irregular surfaces demand maximum limb flexibility. The floating shoulder architecture distributes impact forces across musculature rather than skeletal joints, reducing injury risk during the high-impact landings that characterize feline hunting strategies. Riding reflex and vestibular sophistication. The cat's ability to rotate its body midair and land on its feet emerges from one of the most sophisticated vestibular systems in the animal kingdom. Within milliseconds of beginning a fall, the feline brain receives orientation data from the inner ear's semicircular canals and processes this information to initiate the riding reflex. The sequence begins with the head rotating toward the upright position, followed by the spine twisting, the front legs aligning, and finally the hindquarters completing the rotation. This occurs with such precision that cats can right themselves from a fall initiated upside down in a space as short as 30 centimeters. The physics of this rotation defies intuitive conservation of angular momentum because cats accomplish the maneuver with zero initial angular velocity. They achieve this by extending their legs on one side while tucking them on the other, creating a differential moment of inertia that allows rotation without violating physical laws. The flexible, nearly 40 vertebrae spine acts as a physiological gimbal, permitting front and rear body segments to rotate semi-independently. Interestingly, cats reach terminal velocity at approximately 60 miles per hour, and falls from intermediate heights often prove more dangerous than falls from greater heights, as the cat hasn't had time to fully complete the riding sequence and adopt the spread limb parachute posture that reduces impact force. Fast twitch muscle predominance. The feline muscular system demonstrates extreme specialization toward explosive power rather than endurance, with approximately 90% of muscle fibers classified as fast twitch or type 2 fibers. These fibers contract rapidly and generate substantial force but fatigue quickly, creating an animal capable of extraordinary athletic feats sustained only briefly. A domestic cat can accelerate to 30 miles per hour in short bursts, leap vertically more than 5 times its height, and rotate its body midair with precision that appears to defy physics. However, these remarkable performances come at a metabolic cost that limits sustained activity. The preponderance of fast-twitch fibers reflects the feline hunting strategy of patience followed by explosive ambush rather than the endurance pursuit practiced by canids. This physiological profile influences behavior profoundly, Cats in the wild spend approximately 18 hours daily in rest or sleep, conserving energy for the brief, intense predatory sequences that secure their meals. The muscle fiber composition also contributes to the characteristic fluidity of feline movement, as fast-twitch fibers enable the rapid, precise adjustments that create the impression of liquid motion. Even domestic cats retain this physiology unchanged, explaining why play sessions exhaust cats quickly and why they instinctively structure their activity in brief, intense episodes separated by extended rest periods. Grooming and papillae architecture. The feline tongue represents a marvel of biological engineering, covered with hundreds of rear-facing, hollow-tipped spines called filiform papillae. Recent high-resolution imaging has revealed that these structures, composed of keratin arranged in a scoop-like shape, function as miniature capillary tubes. During grooming, the papillae penetrate through the fur to reach the skin, where they draw up saliva through capillary action and deposit it on the epidermis. This delivery system enables cats to clean skin layers that would be unreachable with smooth tongues, while the mechanical action of the papillae removes loose fur, debris, and parasites with remarkable efficiency. A single grooming session can transfer up to quarter of the cat's body weight in saliva to its coat, which then evaporates and provides evaporative cooling, critical for an animal with minimal sweat glands. The papillae's rear-facing orientation also explains why objects caught on a cat's tongue become increasingly entangled with struggle. Each papilla is anchored in a flexible base that allows it to pivot, creating a self-cleaning mechanism when the cat closes its mouth. The entire tongue surface acts as a distributed pump, capable of moving significant fluid volumes through its hierarchical capillary structure with minimal energy expenditure, representing elegant biomechanical optimization. Obligate carnivore metabolism. Cats stand nearly alone among domestic animals as obligate carnivores, meaning they cannot synthesize several amino acids and vitamins essential for survival from non-animal sources. Unlike omnivores that can convert beta-carotene to vitamin A or synthesize taurine from other amino acids, cats must obtain these nutrients directly from animal tissue. The evolutionary loss of these metabolic pathways reflects millions of years of consistent carnivory, where selective pressure favored efficiency over metabolic redundancy. The feline liver lacks significant glucokinase activity, the enzyme that processes dietary carbohydrates, and instead maintains blood glucose through gluconeogenesis from amino acids even when carbohydrates are available. This metabolic inflexibility means cats convert dietary protein to glucose regardless of macronutrient ratios, making them unable to spare protein even when fed high-carbohydrate diets. Taurine deficiency causes retinal degeneration and dilated cardiomyopathy in cats, conditions rarely seen before commercial cat foods began supplementation. The requirement for arachidonic acid, a fatty acid cats cannot synthesize from linoleic acid, further illustrates this metabolic dependence on animal tissue. These biochemical constraints define not only dietary needs but also influence hunting behavior, territorial requirements, and social structure, as cats must secure regular access to animal prey for survival rather than being able to supplement with vegetation during resource scarcity. Territorial marking and chemical landscapes. Cats inhabit a world rich in chemical signals that remain largely imperceptible to humans, constructing elaborate olfactory territories through strategic deposition of scent marks. The sebaceous glands concentrated on the face, particularly around the lips, chin, and temporal regions, produce complex lipid-based secretions that cats transfer to objects through head rubbing. These facial pheromones convey individual identity and establish familiarity rather than territorial claims. In contrast, urine marking and anal gland secretions contain volatile compounds that assert territorial ownership and convey reproductive status across distances and time. Male cats produce feline in their urine, a sulfur-containing amino acid that degrades into characteristic male cat odor compounds over time, creating a temporal signature that reveals how recently a location was marked. Scratching behavior serves dual purposes: visual marking through the physical scratch pattern and olfactory marking through interdigital scent glands between the toe pads. Research has revealed that cats can discriminate between their own scent marks and those of conspecifics, and they preferentially refresh their own marks while investigating novel scents extensively. The spatial distribution of scent marks creates a cognitive map of territorial boundaries and core areas, with marking density highest in transitional zones where territories overlap. The genetics of coat color and pattern. The seemingly infinite variety of feline coat colors and patterns emerges from a relatively simple genetic foundation involving fewer than a dozen key loci. The basic color genes, black, brown, and orange, interact with dilution factors, white spotting genes, and pattern modifiers to produce the spectrum of feline appearances. The orange coat color gene, located on the X chromosome, creates the phenomenon of tortoiseshell and calico cats, which are almost exclusively female due to exon activation patterns during development. Each cell in a female cat carrying one orange and one non-orange allele randomly inactivates one X chromosome, creating the characteristic mosaic pattern where patches of orange and black, or their diluted versions, appear adjacent to each other. The tabby pattern, considered the ancestral wild-type pattern, results from the agoutis signaling protein gene, which creates banded coloration on individual hairs. Different alleles at the tabby locus produce mackerel, classic, spotted, or ticked patterns through variations in when and where pigment production occurs during hair growth. The dominant white gene, distinct from albinism, masks underlying color genetics and frequently causes deafness through degeneration of the cochlear structures during development, particularly in blue-eyed white cats where the penetrance of deafness approaches 75%. Feline hearing and ultrasonic perception. The feline auditory system operates across a frequency range extending from 48 Hz to 85 kHz, granting cats access to acoustic worlds entirely beyond human perception. This upper limit, nearly 2 octaves above human hearing capacity, allows cats to detect the ultrasonic vocalizations of rodent prey, which communicate among themselves at frequencies humans cannot hear. The external pinnae, those mobile ear flaps composed of 32 muscles, can rotate independently through 180 degrees, functioning as acoustic radar dishes that localize sound sources with precision approaching 5 degrees of accuracy. The ear's orientation provides the brain with differential time of arrival and intensity cues that enable three-dimensional sound localization. Within the cochlea, the organ of Corti contains approximately 45,000 hair cells arranged along a basilar membrane that resonates at different positions according to frequency, with the high-frequency regions disproportionately developed compared to other mammals. This cochlear specialization reflects the evolutionary importance of detecting high-frequency sounds produced by small prey. Interestingly, cats show reduced sensitivity in the human vocal frequency range, which may explain why they often appear indifferent to verbal communication. The auditory cortex dedicates substantial processing resources to temporal patterns and rapid frequency modulation, enabling cats to distinguish between sounds separated by intervals as brief as 6 milliseconds. Ancient Egyptian domestication and deity status. The symbiotic relationship between cats and humans likely began approximately 9,500 years ago in the Fertile Crescent, where genetic evidence points to the Near Eastern wildcat as the primary ancestor of domestic cats. However, the relationship crystallized into something approaching modern domestication in ancient Egypt around 4,000 years ago, where cats became integral to grain storage protection and eventually achieved status as living deities. The Egyptian goddess Bat, originally depicted as a fierce lioness but later as a domestic cat or cat-headed woman, presided over home, fertility, and childbirth, while also protecting the sun god Ra from his serpentine nemesis. Cats in Egypt held legal protection so absolute that killing one, even accidentally, could result in execution. When household cats died, families went into mourning, shaving their eyebrows as a sign of grief, and the deceased cats were mummified and buried with ceremonies rivaling human funerals. The city of Bubastis contained vast cat cemeteries where hundreds of thousands of mummified cats have been excavated. This religious significance stemmed partly from the cat's role in protecting grain stores from rodents and venomous snakes, but also from observed behaviors that seemed to embody divine qualities. Their nocturnal activity and glowing eyes suggested supernatural connection, while their predatory efficiency appeared almost magical. Medieval European persecution and population collapse. The reverence cats enjoyed in ancient Egypt contrasted sharply with their fate in medieval Europe. where they became associated with paganism, witchcraft, and demonic forces. This shift in perception began as Christianity consolidated power and sought to eliminate competing religious practices that honored animals as sacred. By the 13th century, Pope Gregory IX issued a papal bull declaring cats, particularly black ones, as instruments of Satan, which catalyzed systematic persecution across Europe. Cats were captured, tortured, and killed in public spectacles that combined religious fervor with communal entertainment. The celebration of feast days often included throwing living cats from towers, burning them in bonfires, or sealing them into walls during building construction. This sustained campaign of extermination devastated cat populations across the continent precisely when their rodent control services were most critically needed. Contemporary historians have drawn connections between the decline in cat populations and the catastrophic spread of the Black Death in the 14th century. With fewer cats to control rat populations, the fleas carrying Yersinia pestis found ideal conditions for epidemic transmission. While debate continues regarding the magnitude of this effect, the correlation between cat persecution and plague severity remains historically striking, illustrating the ecological consequences of disrupting predator-prey dynamics through cultural practices rooted in superstition. The mechanics of the fatal bite. The killing technique employed by cats demonstrates remarkable neurological sophistication and anatomical precision. When a cat successfully subdues prey, it delivers a bite aimed at the back of the neck, specifically targeting the space between cervical vertebrae. The placement of this bite is not random but guided by extraordinarily sensitive mechanoreceptors embedded throughout the periodontal ligaments surrounding the canine teeth. These sensors can detect subtle variations in tissue density and resistance, allowing the cat to feel the gap between vertebral processes where the spinal cord is most accessible. The canine teeth, being slightly flattened rather than perfectly round in cross-section, are optimized to slide between bones without being deflected. Once positioned correctly, the cat applies force with temporal muscles that generate bite forces exceeding 100 pounds per square inch at the tooth tip despite the cat's relatively small size. The killing bite severs the spinal cord or compresses vital blood vessels to the brain, causing rapid unconsciousness. This technique reflects millions of years of refinement, as inefficient killers would have faced injured, fighting prey that could inflict damage, while cats capable of quick, clean kills minimized risk and energy expenditure while maximizing hunting success. Thermoregulation and heat tolerance. Cats evolved in arid environments where efficient thermoregulation proved essential for survival, developing a physiology that conserves water while managing heat load. Unlike dogs that rely heavily on panting for evaporative cooling, cats possess minimal sweat glands, located only on the paw pads and a few facial locations. This paucity of sweat glands reduces water loss but necessitates alternative cooling strategies. The primary feline thermoregulatory mechanism involves behavioral adaptation, seeking shade, reducing activity during heat peaks, and adopting stretched-out postures that maximize surface area for radiative and convective cooling. When ambient temperature approaches body temperature, cats begin grooming more frequently, spreading saliva across their fur where it evaporates and provides significant cooling. The feline coat itself contributes to thermoregulation through its insulating properties, which paradoxically help in heat by creating a barrier against radiant heat absorption. Cats demonstrate a remarkable tolerance for high body temperatures with their thermoneutral zone, the temperature range where metabolic rate remains minimal, extending up to 95 degrees Fahrenheit. However, this heat tolerance comes with limited capacity for active heat dissipation, making cats vulnerable to hyperthermia when stressed or confined in hot environments, as they lack the panting efficiency that canids employ for rapid heat loss. Feline sleep architecture. Cats spend approximately 2/3 of their lives in sleep, with adult cats sleeping 14 to 18 hours daily, but this rest involves complex sleep architecture distinct from simple unconsciousness. Feline sleep divides into phases of light sleep and deep sleep, with cats spending 75% of sleep time in light, slow-wave sleep where they maintain sufficient muscle tone to respond instantly to stimuli. This state, often accompanied by upright head position and closed eyes with occasional twitching ears, allows rapid transition to wakefulness if threats or opportunities appear. The remaining 25% of sleep time involves rapid eye movement or REM sleep, characterized by complete muscle relaxation except for whisker and paw twitching. During these periods, which typically last only 5 to 7 minutes per cycle, cats likely experience dreams, as suggested by the patterned movements that sometimes accompany REM sleep. The evolutionary driver for this sleep pattern relates to the predatory lifestyle. After the intense energy expenditure of a hunt and the protein-rich meal that follows, extended rest periods allow for digestion and tissue repair while conserving energy. The polyphasic sleep pattern, involving multiple sleep-wake cycles throughout the day and night rather than one consolidated sleep period, reflects the natural hunting rhythm where multiple hunting opportunities occur throughout each 24-hour period. Retractable claws and digital mechanics. The retractable claw represents a defining feature of most felids, achieved through an elegant anatomical arrangement involving elastic ligaments, digital bones, and flexor tendons. In the resting position, dorsal elastic ligaments pull the distal phalanx upward and backward, retracting the claw into a protective sheath and keeping it elevated off the ground during locomotion. This default retracted position keeps claws sharp by preventing contact with abrasive surfaces while moving, preserving their effectiveness for climbing, fighting, and prey capture. When needed, the digital flexor muscles contract, pulling the tendon that forces the distal phalanx downward and forward, extending the claw and creating a formidable weapon or climbing tool. The mechanics involve a three-bone system, the proximal, middle, and distal phalanges, articulated to allow the tip segment to rotate nearly 90 degrees from retracted to extended positions. Cheetahs represent the notable exception among felids, possessing only semi-retractable claws that remain partially extended to provide traction during high-speed pursuits, a specialization that sacrifices climbing ability for running performance. The domestic cat's claws grow continuously throughout life at approximately 1 millimeter per week, requiring regular scratching behavior to remove the worn outer sheath and expose the sharp new claw beneath. This maintenance behavior also serves marking functions and provides crucial stretching exercise for the digital flexor muscles. Olfactory cortex and scent processing. While cats are typically celebrated for their visual and auditory capabilities, their olfactory system demonstrates sophistication rivaling that of specialist scent hunters. The feline nose contains approximately 65 to 80 million olfactory receptor cells, nearly twice the number found in humans, distributed across an olfactory epithelium with surface area expanded by complex turbinate structures. These receptors connect to the olfactory bulb, a neural structure disproportionately large relative to brain size, occupying nearly 5% of total brain volume. The olfactory cortex processes scent information through both primary pathways that provide conscious smell perception and accessory pathways connecting to the amygdala and hypothalamus that influence emotional and hormonal responses without conscious awareness. This dual processing explains how scents can alter feline behavior dramatically without the cat appearing to consciously notice the odor. Cats possess approximately 200 million olfactory receptor genes, creating a combinatorial system capable of discriminating between thousands of distinct odors and detecting certain compounds at concentrations below 1 part per trillion. The turbinate bones create turbulent airflow through the nasal cavity, increasing contact between air and sensory epithelium while warming and humidifying incoming air. Interestingly, cats avoid citrus scents universally, suggesting that smell aversion may have genetic components linked to protective mechanisms against potentially toxic plant compounds. The flamen response and pheromone detection. When a cat encounters particularly intriguing scents, especially those of biological significance like urine marks from other cats, it often performs a distinctive behavior called the flamen response, characterized by mouth opening, lip curling, and a momentary expression of apparent disgust or intense concentration. This behavior facilitates the transfer of odor molecules from the mouth to the vomeronasal organ through two small ducts located just behind the upper incisors called the incisive papillae. By drawing air into the mouth and pressing the tongue against the palate, cats create a pumping action that moves chemical-laden air through these ducts into the vomeronasal organ. This specialized chemosensory structure contains receptor neurons genetically distinct from those in the main olfactory epithelium, specifically tuned to detect pheromones and other non-volatile biological signals. The vomeronasal system projects directly to brain regions controlling reproductive and social behavior, enabling chemical messages to trigger behavioral changes without cortical processing. Male cats exhibit flamen responses more frequently than females, particularly when investigating female urine for reproductive status indicators. The behavior peaks during breeding season and diminishes in neutered animals, suggesting hormonal modulation of this sensory behavioral circuit. Recent research has identified specific proteins in cat urine called feline urinary proteins that bind and stabilize volatile pheromones, extending their environmental persistence and enhancing their biological effect when detected through the vomeronasal organ. Hunting sequence and predatory motor patterns. The stereotyped sequence of behaviors cats employ during hunting reveals fixed action patterns shaped by millions of years of predatory evolution. The hunting sequence typically progresses through distinct phases: orientation, stalking, pouncing, capture, and killing, each governed by specific neural circuits that activate in response to particular stimuli. The sequence begins when movement in the visual periphery triggers orientation responses, the head turns, ears rotate toward the stimulus, and the cat assumes a frozen, watchful posture. If the stimulus continues to exhibit prey-like characteristics, small size, rapid movements, attempts to flee, the stalk phase initiates. During stalking, cats adopt a characteristic crouched posture with belly low and hindquarters elevated, advancing with slow, deliberate steps punctuated by frozen pauses. The body remains in constant low-level tension, with subtle weight shifting maintaining balance while minimizing detectable movement. As distance closes, the pounce sequence activates. Hindquarters wiggle slightly as weight shifts rearward, muscles load like compressed springs, then release in an explosive leap directed at the projected intercept point. Capture involves forelimb extension with claws extended, grasping the prey and bringing it toward the mouth for the killing bite. These motor patterns are so deeply embedded that they emerge in kittens without prior experience and appear during play with inanimate objects, demonstrating their genetic basis rather than learned nature. Kitten development and sensory maturation. Kittens are born remarkably underdeveloped compared to precocial species, emerging with closed eyes, folded ear pinnae, and limited locomotor abilities. This altricial state reflects an evolutionary trade-off where shortened gestation periods allowed maternal flexibility while necessitating extended postnatal development. The eyes open between 7 and 14 days after birth, but visual acuity remains poor for several additional weeks as retinal structures mature and neural connections refine. The ear canals open around the same time, but sophisticated auditory processing capabilities don't emerge until the 3rd week. Interestingly, kittens can purr from birth despite their overall developmental immaturity, suggesting this behavior serves crucial early functions, likely in maternal offspring communication and possibly in stimulating milk production. The critical socialization period extends from approximately 2 to 7 weeks of age, during which kittens form lasting impressions about social partners, environmental features, and appropriate fear responses. Exposure to humans during this window proves essential for developing friendly relationships with people, while isolation during this period often results in permanently feral behavior patterns resistant to later socialization efforts. Motor development proceeds in characteristic stages, with walking emerging around the 3rd week, running and playing by the 4th week, and adult-like coordination achieved by the 8th week. The retractable claw mechanisms don't become fully functional until approximately 4 weeks, protecting young kittens from self-injury during uncoordinated movements. The cerebellum and movement coordination. The feline cerebellum, the hindbrain structure responsible for movement coordination and motor learning, occupies a proportionally larger volume relative to overall brain size than in most other mammals. This neural investment reflects the extraordinary precision required for feline predatory success, where miscalculations of millimeters during a pounce mean the difference between capture and escape. The cerebellum receives input from proprioceptive sensors throughout the body, visual and vestibular systems, and motor cortex commands, integrating this multisensory information to generate smooth, coordinated movements and real-time corrections. During a jump, the cerebellum calculates trajectory, orchestrates the sequence of muscle contractions, and makes micro-adjustments during flight based on vestibular feedback. The structure enables cats to perform ballistic movements, actions completed too rapidly for sensory feedback to influence them, with remarkable accuracy by maintaining internal models of body mechanics and external physics. These forward models predict the sensory consequences of motor commands, allowing preemptive corrections before errors become apparent. The climbing abilities that make cats so distinctively feline depend on cerebellar computations that maintain balance on narrow surfaces, coordinate independent limb movements on irregular substrates, and calculate dynamic gripping forces. Damage to feline cerebellar structures produces characteristic deficits including intention tremors, ataxia, and inability to judge distances, highlighting the structure's essential role in the fluid movement that defines the feline form. Catnip response and nepetalactone sensitivity. The distinctive behavioral response cats display toward catnip, nepetacataria, results from volatile terpenoid compounds, particularly nepetalactone, which bind to olfactory receptors and trigger neurological responses mediated through the olfactory system. Approximately 50 to 75% of cats display sensitivity to catnip, with the response controlled by an autosomal dominant gene that determines whether the cat possesses the relevant receptors. Affected cats exhibit a stereotyped behavioral sequence upon exposure: initial sniffing and licking, head shaking and chin rubbing, body rolling, and often vocalization, followed by a refractory period lasting approximately 2 hours during which the cat shows no response to additional exposure. The behavior pattern resembles aspects of female sexual behavior, suggesting that nepetalactone may activate neural circuits related to reproductive responses, though both males and females respond equally. Recent studies have revealed that nepetalactone also functions as an insect repellent, particularly against mosquitoes, and cats that respond to catnip show increased grooming and rubbing behaviors that effectively transfer the compound to their fur, potentially conferring protection against biting insects. This suggests the response may have evolutionary significance beyond mere recreational pleasure. Interestingly, many wild felid species including lions, tigers, and leopards also respond to catnip, indicating this sensitivity predates the evolution of domestic cats and serves some adaptive function, although its precise evolutionary advantage remains debated among researchers. Polydactyl cats and Hemingway's colony. A naturally occurring genetic mutation causes polydactyly in cats, resulting in extra digits on one or more paws through a dominant autosomal gene that disrupts normal limb development. While typical cats possess 18 toes total, 5 on each front paw and 4 on each rear paw, polydactyl cats may have up to 7 digits per paw, with the most common pattern showing extra thumbs on the front feet. These additional digits often appear more fully formed than the dewclaw, sometimes including multiple bones and functional movement. The mutation reached cultural prominence through writer Ernest Hemingway, who received a polydactyl cat named Snow White from a ship captain in the 1930s. This founding cat established a colony at Hemingway's Key West home that persists today, with approximately half the 50 resident cats displaying polydactyly. Maritime folklore held that polydactyl cats, common among ship cats of the 19th century, brought good luck and possessed superior hunting abilities, with their extra toes supposedly providing better balance on pitching decks. Genetic studies have revealed that the polydactyly mutation arose independently multiple times in different cat populations, suggesting the trait doesn't impose significant survival disadvantages and may even offer advantages in certain contexts. Some polydactyl cats demonstrate increased dexterity with their extra digits, manipulating objects in ways resembling thumb opposition, though true opposable thumbs remain beyond current feline capabilities. The trait continues to intrigue geneticists as a model for understanding limb patterning and developmental genetics. Feline immunodeficiency virus and retroviral evolution. Feline immunodeficiency virus represents a lentivirus closely related to human immunodeficiency virus, offering crucial insights into retroviral evolution and immune system interactions. FIV infects domestic cats worldwide, transmitted primarily through deep bite wounds that introduce infected saliva directly into the bloodstream. The virus targets CD4+ T lymphocytes, macrophages, and other immune cells, gradually depleting immune function over years in a progression remarkably similar to HIV in humans. However, significant differences exist. FIV-infected cats often survive for years without developing severe immunodeficiency, and the virus shows restricted host specificity, failing to cross species barriers readily. This species specificity makes FIV non-infectious to humans, allowing researchers to study lentiviral pathogenesis in its natural host without zoonotic risk. The viral genome reveals evolutionary relationships suggesting ancient origins, with evidence that related lentiviruses have infected felid species for millions of years, leading to coevolution between virus and host immune systems. Some wild felid species carry FIV strains without apparent disease, suggesting either ancient coevolution or inherent resistance mechanisms. Domestic cat FIV infection proceeds through acute, asymptomatic, and terminal phases spanning 5 to 10 years typically, though considerable individual variation exists. The virus integrates into host chromosomes as a provirus, making elimination effectively impossible with current technology. Research on FIV vaccine development and therapeutic interventions has contributed significantly to understanding retroviral biology, with findings often applicable to human HIV research. Toxoplasmosis and the feline definitive host. Toxoplasma gondii, a protozoan parasite of profound epidemiological importance, completes its sexual reproduction cycle exclusively within feline intestinal epithelium, making cats the definitive hosts in a complex life cycle. Infected cats shed environmentally resistant oocysts in feces for 1 to 3 weeks following initial infection, typically during kittenhood when the immune system hasn't yet developed protective responses. These microscopic oocysts sporulate in the environment over 1 to 5 days, becoming infectious to virtually all warm-blooded animals including birds, rodents, and humans. Intermediate hosts develop tissue cysts containing dormant bradyzoites that persist for the host's lifetime, particularly in neural and muscular tissue. When cats consume infected prey, the cycle completes. The parasite demonstrates remarkable neurotropism, with tissue cysts showing preference for brain regions including the amygdala. Research has revealed that infected rodents display reduced fear of cat odors and increased exploratory behavior, alterations that increase predation risk and enhance parasite transmission to its definitive host. This behavioral manipulation suggests sophisticated evolved mechanisms for parasite transmission enhancement. In humans, approximately 30% of the global population carries latent toxoplasmosis, generally causing no symptoms in immunocompetent individuals but potentially influencing subtle behavioral patterns and reaction times, though the significance of these effects remains debated. Congenital transmission poses the primary human health concern, as infection during pregnancy can cause severe fetal abnormalities. The Jacobson's organ in chemical analysis. The vomeronasal organ's function extends beyond simple pheromone detection, serving as a sophisticated chemical analysis system capable of extracting complex information from biological samples. The organ consists of paired tubular structures lined with sensory epithelium containing chemoreceptor neurons distinct from those in the main olfactory system. These neurons express vomeronasal receptors from the V1R and V2R gene families, with cats possessing approximately 30 V1R genes and 9 V2R genes, creating a combinatorial receptor repertoire capable of discriminating between hundreds of chemical signatures. The vomeronasal neurons send axonal projections to the accessory olfactory bulb, which in turn projects primarily to the medial amygdala and hypothalamus rather than the olfactory cortex, creating a neural pathway that influences emotional and reproductive behavior without conscious perception. This anatomical arrangement enables chemical signals to alter behavioral states directly, bypassing cognitive processing. The system shows particular sensitivity to sulfur-containing compounds, proteins, and peptides, precisely the chemical signatures found in urine and other biological secretions that convey individual identity, reproductive status, and health information. Recent research has demonstrated that vomeronasal detection of male pheromones can trigger neuroendocrine responses in female cats, advancing reproductive cycling and increasing receptivity, illustrating direct chemosensory control over reproductive physiology. The precision of this chemical communication system rivals visual or auditory signals in information content while providing temporal persistence that other sensory modalities cannot match. Cardiac anatomy and feline cardiomyopathy. The feline heart demonstrates anatomical features adapted for an athletic lifestyle requiring periodic explosive exertion, with relatively large ventricular chambers and thick muscular walls capable of generating the high cardiac outputs needed for sprint hunting. Normal feline heart rates range from 140 to 220 beats per minute at rest, approximately double human resting heart rates, reflecting the higher metabolic rate characteristic of small mammals. However, domestic cats suffer from hypertrophic cardiomyopathy at rates exceeding 15% in some populations, making it the most common feline cardiac disease. This condition involves thickening of the left ventricular wall, reducing chamber volume and compromising filling during diastole. The disease shows familial clustering in certain breeds including Maine Coons and Ragdolls, where specific mutations in genes encoding sarcomeric proteins have been identified. The MYBPC3 gene mutation in Maine Coons produces a truncated cardiac myosin-binding protein C, disrupting normal sarcomere assembly and triggering compensatory hypertrophy. Clinical manifestations include heart failure, arterial thromboembolism, and sudden death, often with minimal prior symptoms. The prevalence of this genetic disease raises questions about breeding practices and whether selection for certain physical traits inadvertently concentrated deleterious cardiac mutations. Echocardiography reveals subclinical disease in many cats, suggesting broader genetic influences on cardiac development and function within feline populations than previously recognized. Feline cognition and object permanence. Cognitive studies reveal that cats possess sophisticated mental representation abilities, including object permanence, the understanding that objects continue to exist even when not directly perceived. Experimental paradigms involving hidden objects demonstrate that cats search systematically for concealed items and show surprise when hidden objects disappear or change location in violation of physical laws. These abilities emerge during kitten development around 8 weeks of age, following a progression similar to that observed in human infant cognitive development. Cats demonstrate means-end understanding, using tools appropriately to obtain rewards and showing flexibility when familiar methods fail. Social cognition studies indicate cats distinguish familiar humans from strangers through multiple sensory modalities and recognize their own names even within strings of similar-sounding words, though they often choose not to respond, a behavior sometimes mistaken for cognitive limitation but actually reflecting motivational factors. Working memory tests show cats can retain information about hidden locations for at least 60 seconds and possibly longer, though performance degrades with interference. Problem-solving studies reveal that cats employ trial-and-error learning but also demonstrate insight learning where solutions appear suddenly after periods of apparent inactivity, suggesting mental simulation of solutions before attempting them. The cognitive abilities of cats appear adapted for solitary hunting requirements rather than social cooperation, resulting in a profile emphasizing spatial memory, physical causality understanding, and independent decision-making rather than the cooperative and communicative cognitive strengths emphasized in species like dogs. Whisker fatigue and sensory overstimulation. The term whisker fatigue, though physiologically imprecise, describes a genuine phenomenon where overstimulation of the mechanoreceptors surrounding whisker follicles produces stress responses in cats. Each whisker follicle contains between 100 and 200 mechanoreceptor neurons that fire action potentials with each whisker deflection, creating a continuous stream of sensory information processed by dedicated columns in the somatosensory cortex. Unlike most sensory systems that show adaptation, reduced response to sustained stimulation, the whisker system maintains high sensitivity across extended periods, meaning prolonged or repeated stimulation doesn't diminish significantly. When cats feed from narrow bowls or engage in activities requiring sustained whisker contact with surfaces, the continuous barrage of sensory input may overwhelm processing capacity or trigger stress responses through unknown mechanisms. Affected cats may show food avoidance, irritability, or preference for feeding from wide, flat dishes that minimize whisker contact. The phenomenon intersects with research on sensory processing sensitivity, where individuals, both human and animal, show heightened reactivity to sensory input. Some cats appear more susceptible than others, suggesting individual variation in sensory processing thresholds. While research specifically investigating whisker fatigue remains limited, broader studies of feline stress responses support the concept that sensory environment significantly impacts feline welfare. The practical implications encourage environmental modifications that reduce unnecessary whisker stimulation, particularly during feeding, grooming, and veterinary examination, potentially improving welfare through simple adjustments to how humans structure feline environments. Tail communication and visual signaling. The feline tail serves multiple functions beyond the obvious role in balance, acting as a sophisticated communication device broadcasting emotional state and social intentions through position, movement patterns, and piloerection. The tail contains approximately 23 vertebrae, depending on breed and individual variation, each articulated to allow a remarkable range of motion. Neural control pathways enable independent segments of the tail to move in different patterns simultaneously, creating complex signals. The vertical tail with tip curved forward signals friendly approach intentions and confidence, while a fully vertical tail with quivering motion indicates intense greeting excitement, particularly toward familiar humans. The horizontally extended tail with low positioning communicates uncertainty or submissive intent, transitioning to fear or defensive aggression as the tail tucks completely between the legs. Rapid side-to-side thrashing indicates agitation, irritation, or predatory excitement, with speed and amplitude correlating to arousal intensity. The bottle brush tail, where piloerection causes fur to stand perpendicular to the skin, combines with arched body posture to signal defensive threat and makes the cat appear larger to potential threats. Interestingly, subtle tail movements during sleep likely reflect dream content, suggesting that even unconscious neural activity activates motor programs for tail signaling. The communicative sophistication of tail movements indicates significant cortical control overlaying reflexive circuits, allowing intentional social signaling rather than merely automatic emotional expression. Breeds with naturally shortened or absent tails show compensatory increases in facial expressions and vocalization, highlighting the tail's importance in the natural feline communication repertoire. The premolar gap and dental specialization. The distinctive gap between the feline canine and premolar teeth, called the diastema, represents an evolutionary adaptation facilitating the carnassial slicing mechanism. This space allows the long canine teeth to extend fully without interference from adjacent teeth while providing room for prey or food material to be positioned optimally for the carnassial shear. The dental formula of cats: 3 incisors, 1 canine, 3 premolars, and 1 molar on each side of the upper jaw, with 3 incisors, 1 canine, 2 premolars, and 1 molar on each side of the lower jaw, reflects progressive loss of teeth from ancestral carnivores. This reduction represents evolutionary refinement toward efficient killing and meat processing rather than omnivorous flexibility. The incisors, small and relatively uniform, serve primarily for grooming and scraping rather than food processing. The prominent canines, slightly recurved and oval in cross-section, are designed for precision piercing and prey restraint rather than crushing. The 4th upper premolar and 1st lower molar form the carnassial pair, with sharp cusps and shearing edges that slide past each other like scissors, cutting rather than grinding. The absence of grinding molars typical of omnivores reflects the fact that cats swallow most food in chunks rather than chewing it thoroughly. This dental arrangement limits cats to head positions that align the carnassials with food items, explaining the distinctive head-tilting behavior observed when cats process larger food pieces. Feline diabetes and pancreatic function. Diabetes mellitus affects cats with increasing frequency in modern populations, with prevalence estimated at approximately 1 in 200 cats. The disease typically presents as type 2 diabetes, characterized by insulin resistance and eventual pancreatic beta cell exhaustion rather than the autoimmune destruction of beta cells seen in type 1 diabetes. The feline pancreas contains islets of Langerhans distributed throughout the organ, with beta cells producing insulin and alpha cells secreting glucagon. In diabetic cats, amyloid deposits accumulate in these islets, disrupting normal architecture and contributing to beta cell dysfunction. Obesity represents the primary risk factor, as excess adipose tissue secretes inflammatory cytokines and adipokines that promote insulin resistance in peripheral tissues. The obligate carnivore metabolism of cats creates unique challenges in managing diabetes, as the glucose-independent protein metabolism continues regardless of insulin availability, preventing the simple dietary modifications effective in omnivorous species. Treatment typically requires exogenous insulin administration, usually a long-acting form given twice daily subcutaneously. Interestingly, some diabetic cats achieve remission, restoration of normal glycemia without insulin therapy, particularly when diabetes is diagnosed early and treated aggressively. This occurs more frequently in cats than in humans, possibly reflecting differences in pancreatic reserve capacity or the reversibility of lipotoxic effects on beta cells. The rising incidence of feline diabetes parallels increasing obesity rates in pet cat populations, suggesting that domestication's metabolic mismatches contribute significantly to this modern feline health challenge. Chromosomal architecture and feline genetics. The domestic cat genome, sequenced completely in 2007, contains approximately 2.7 billion base pairs organized across 19 chromosomal pairs, revealing evolutionary relationships and disease susceptibilities. The relatively compact genome size compared to many mammals reflects efficient genome organization with minimal repetitive DNA. Comparative genomics reveals that cats diverged from other carnivores approximately 10.8 million years ago, with the Felidae family showing relatively recent radiation. Most extant felid species separated less than 11 million years ago, making the family among the most recently evolved mammalian groups. This recent divergence explains the high degree of genetic similarity between domestic cats and wild felids, with hybridization remaining possible between domestic cats and several wild species including European wildcats and African wildcats. The feline X chromosome carries numerous genes beyond those involved in sex determination, including the orange coat color locus and several genes related to blood clotting factors. This X linkage explains various sex-linked traits and disorders observed in cat populations. The cat genome contains approximately 20,000 protein-coding genes, fewer than human but comparable to most mammals. Interestingly, cats possess reduced genetic diversity compared to dogs, likely reflecting a smaller founding population during domestication and less intensive artificial selection historically. The genome sequence has facilitated identification of genetic mutations underlying inherited diseases, enabling development of genetic tests for conditions including hypertrophic cardiomyopathy, polycystic kidney disease, and progressive retinal atrophy, feline leukemia virus, and oncogenesis. Feline leukemia virus, a gamma-retrovirus first isolated in the 1960s, represents a major infectious disease affecting domestic cats worldwide despite effective vaccines. The virus transmits through saliva, nasal secretions, and maternal transfer, requiring close contact for spread unlike the respiratory droplet transmission of many viral diseases. Following infection, cats proceed through several possible outcomes. Some eliminate the virus completely through effective immune responses within weeks. Others develop persistent viremia with viral replication continuing indefinitely, while a third group becomes latently infected with viral DNA integrated into host chromosomes but no active replication. Persistently infected cats typically succumb to FALV-related diseases within 3 years of diagnosis, though this has improved with modern veterinary care. The virus causes pathology through multiple mechanisms. Direct oncogenic transformation through insertional mutagenesis, where viral DNA integration disrupts tumor suppressor genes or activates oncogenes, immunosuppression rendering cats susceptible to secondary infections, and bone marrow suppression causing anemias and other cytopenias. The virus contributed significantly to understanding retroviral oncogenesis before the discovery of HIV, with research revealing how retroviruses can transform normal cells into tumor cells. FALV was among the first retroviruses shown to cause cancer in natural host populations rather than through laboratory manipulation. Several FALV strains exist with different pathogenic properties, classified into subgroups A, B, and C based on receptor usage and disease manifestations, adding complexity to the infection dynamics and disease progression patterns observed in affected cat populations. The writing reflex neural circuitry. The neural control of the feline writing reflex involves extraordinarily rapid sensory processing and motor coordination, beginning with vestibular system activation within milliseconds of a fall's initiation. The vestibular apparatus, located in the inner ear, contains 3 semicircular canals oriented in perpendicular planes that detect rotational acceleration and 2 otolith organs, the utricle and saccule, that sense linear acceleration and head position relative to gravity. Hair cells within these structures bend in response to fluid movement or otoconia displacement, generating action potentials that travel via the vestibular nerve to brainstem nuclei. These nuclei project to the spinal cord, cerebellum, and visual system, creating integrated neural circuits that coordinate the writing response. The sequence begins with head rotation controlled by neck muscles responding to vestibular input, bringing the head upright first regardless of body orientation. Visual input supplements vestibular information once the eyes orient toward the ground. The cerebellum coordinates the complex sequence of truncal twisting and limb positioning that follows head rotation, with different body segments rotating sequentially due to the flexible spine's segmental control. Proprioceptive feedback from muscle spindles and joint receptors provides continuous information about body configuration, allowing real-time adjustments. The entire sequence, from fall initiation to landing preparation, completes in less than 1 second, demonstrating remarkable neural processing speed. This reflex appears largely innate, emerging in kittens by 3 to 4 weeks of age, though coordination improves with maturation and experience, suggesting both hardwired circuits and experience-dependent refinement. Feline hypertension and renal disease. Systemic hypertension affects approximately 20% of cats over 8 years of age, with prevalence increasing dramatically in cats with concurrent chronic kidney disease. The feline cardiovascular system maintains blood pressure through integrated control involving the sympathetic nervous system, renin-angiotensin-aldosterone axis, and local vascular regulatory mechanisms. Chronic kidney disease disrupts these regulatory systems through multiple pathways. Reduced renal mass decreases the kidney's capacity to excrete sodium, leading to volume expansion. Impaired production of vasodilatory prostaglandins and nitric oxide shifts the balance toward vasoconstriction. And activation of the renin-angiotensin system in response to decreased renal perfusion creates a pathological cycle where hypertension accelerates kidney damage. Feline hypertension often remains clinically silent until severe complications develop, including retinal hemorrhage and detachment causing sudden blindness, hypertensive encephalopathy causing seizures or altered mentation, left ventricular hypertrophy predisposing to heart failure, and accelerated progression of chronic kidney disease. Blood pressure measurement in cats requires consideration of white coat hypertension, where stress elevates readings falsely, necessitating multiple measurements in quiet environments. Treatment typically employs amlodipine, a calcium channel blocker that dilates arterioles and reduces afterload, though angiotensin-converting enzyme inhibitors provide additional benefits through effects on intraglomerular pressure. The high prevalence of hypertension in aging cats reflects both the frequency of underlying kidney disease and possibly fundamental aspects of feline cardiovascular physiology that predispose to hypertension development. Feline infectious peritonitis and coronavirus mutation. Feline infectious peritonitis represents one of the most enigmatic and devastating feline infectious diseases, resulting from mutations in the ubiquitous and typically benign feline enteric coronavirus. The enteric coronavirus infects most cats at some point, usually causing mild or inapparent gastrointestinal infection with fecal shedding that maintains the virus in cat populations. Within individual infected cats, random mutations occasionally transform the enteric coronavirus into a systemic virulent strain that leaves the intestinal tract, infects macrophages, and disseminates throughout the body. The mutation events involve viral spike protein changes that alter cellular tropism from intestinal epithelium to monocytes and macrophages. Once macrophages become infected, they transport virus systemically, triggering severe immune-mediated inflammation rather than effective viral clearance. The disease manifests in two primary forms: effusive or wet FIP, characterized by peritoneal and pleural fluid accumulation with high protein content, and non-effusive or dry FIP, presenting with granulomatous inflammation affecting various organs, particularly the eyes, central nervous system, and abdominal organs. Both forms prove nearly always fatal historically, though recently developed antiviral therapies targeting the viral protease show promise. The immune response paradoxically drives pathology, with antibody-dependent enhancement where virus antibody complexes enter macrophages more efficiently than naked virus, and cell-mediated immune responses creating destructive granulomas and vasculitis. Young cats and cats in high-density environments face elevated risk, likely reflecting both immunological naivety and increased viral exposure, facilitating the rare mutation events that generate virulent strains. Acromelanism and temperature-sensitive coloration. The distinctive pointed pattern observed in Siamese, Himalayan, and related breeds results from a temperature-sensitive mutation in the tyrosinase enzyme required for melanin synthesis. The mutant enzyme retains partial function at cooler temperatures but becomes unstable and non-functional at normal feline body temperature. Consequently, color develops only on cooler extremities, ears, face, paws, and tail, while the warmer core body regions remain pale. Kittens are born entirely white because the uniform warmth of the uterine environment prevents pigment production anywhere on the body. As kittens mature and begin thermoregulating independently, their extremities cool sufficiently for melanin production to activate, with point coloration developing gradually over the first weeks and months of life. The temperature threshold for enzyme function falls around 95 degrees Fahrenheit, explaining why Siamese cats living in warm climates show lighter point coloration than those in cooler environments. The mutation affects an enzyme in the melanin synthesis pathway, specifically the conversion of tyrosine to melanin precursors, resulting in temperature-dependent pigmentation. Injuries causing inflammation and local temperature increases can temporarily alter coat color in these breeds, with shaved areas growing back darker due to the cooler skin temperature during regrowth. The pointed pattern associates with additional phenotypic effects including blue eye color and increased vocalization tendency, suggesting pleiotropic effects of the genes involved or tight genetic linkage between these traits during breed development. Feline mammary tumors and neoplasia. Mammary tumors constitute the third most common neoplasm in female cats, with approximately 85% proving malignant, a dramatically higher malignancy rate than the 50% observed in canine mammary tumors. These carcinomas typically affect older intact or late-spayed female cats, with risks strongly associated with hormonal exposure history. Cats spayed before 6 months of age show 91% reduction in mammary tumor risk compared to intact cats, while spaying after 1 year provides no significant protection, indicating a critical window during mammary development when hormonal influences determine future cancer susceptibility. Feline mammary carcinomas demonstrate aggressive biological behavior with rapid local invasion, high metastatic rates affecting lungs and regional lymph nodes, and poor prognosis overall. Tumor size at diagnosis proves the most significant prognostic factor, with tumors larger than 3 centimeters conferring median survival times measured in months even with aggressive treatment. The molecular pathogenesis involves multiple genetic alterations including mutations in tumor suppressor genes like p53 and activation of growth factor signaling pathways. Unlike human breast cancer, feline mammary tumors rarely express estrogen or progesterone receptors, limiting hormonal therapy options that prove effective in human oncology. Treatment primarily involves surgical excision with radical mastectomy showing better outcomes than conservative lumpectomy, though distant metastases already present at diagnosis frequently limit long-term survival regardless of local tumor control. The high malignancy rate and aggressive behavior of feline mammary tumors underscore the importance of early spaying as a preventive measure and careful monitoring of mammary tissue in older intact or late-spayed females. The nictitating membrane and ocular protection. Cats possess a third eyelid, the nictitating membrane or palpebra tertia, located in the medial corner of each eye that can sweep across the corneal surface providing protection and moisture distribution. This thin, translucent membrane contains cartilage providing structural support and lymphoid tissue contributing to local immune defense. The membrane normally rests retracted in the medial canthus, becoming visible only during partial closure of the primary eyelids or when the eye retracts into the orbit. Movement is passive rather than actively controlled, resulting from retractor bulbi muscle contraction that pulls the globe deeper into the orbit, allowing the nictitating membrane to advance across the corneal surface through passive mechanisms. The membrane distributes tear film across the cornea during its sweeping motion, contributing to the maintenance of the precorneal tear film essential for optical clarity and corneal health. Approximately 30% of the total tear production originates from a lacrimal gland embedded in the nictitating membrane's base. Persistent protrusion of the nictitating membrane, called Haas syndrome or third eyelid elevation, occurs in various disease states including Horner's syndrome, tetanus, dehydration, and systemic illness, serving as a nonspecific indicator of physiological disturbance. The membrane also provides mechanical protection during hunting and fighting, sweeping across the eye when vegetation or struggling prey might cause corneal injury. The evolutionary retention of this structure across felids indicates its continued adaptive value despite the protective advances provided by the primary eyelids. Polycystic kidney disease and genetic screening. Polycystic kidney disease, inherited as an autosomal dominant trait, affects Persian cats and related breeds with prevalence reaching 38% in some populations. The condition results from mutations in the PKD1 gene encoding polycystin 1, a large membrane protein involved in cellular sensing of mechanical stimuli and regulation of tubular epithelial cell proliferation. The mutation causes progressive development of fluid-filled cysts within renal parenchyma, originating from tubular epithelial cells that lose normal growth control. Cysts appear as early as 4 weeks of age in affected kittens, growing progressively throughout life and gradually replacing functional renal tissue. The inexorable cyst expansion eventually causes renal failure, typically in middle age, though considerable variation exists in progression rates between individuals. The disease demonstrates complete penetrance, meaning every cat inheriting the mutation will develop cysts, though clinical severity varies. Ultrasonographic examination can detect cysts in young cats, but genetic testing provides definitive diagnosis even before cysts become detectable, enabling informed breeding decisions. The availability of genetic testing has enabled progressive reduction in disease prevalence through selective breeding programs that exclude carriers. Heterozygous cats cannot be distinguished clinically from homozygous affected cats, but breeding studies suggest homozygosity may cause more severe or earlier onset disease. The PKD1 mutation arose centuries ago within Persian breeding lines and spread through the breed due to popular sire effects where extensively used breeding cats transmitted the mutation to large numbers of offspring, illustrating how genetic bottlenecks in purebred populations can concentrate deleterious alleles. Feline lower urinary tract disease. Feline lower urinary tract disease encompasses a collection of conditions affecting the bladder and urethra, presenting with similar clinical signs including dysuria, polyuria, hematuria, and inappropriate urination. Approximately 60% of cases receive diagnoses of feline idiopathic cystitis, a painful inflammatory condition with unknown etiology showing similarities to human interstitial cystitis. This condition likely involves neurogenic inflammation where stress triggers release of substance P and other neuropeptides from bladder sensory neurons, causing mast cell degranulation, increased vascular permeability, and pain sensitization without infectious or calculogenous cause. Affected cats often display evidence of heightened sympathetic nervous system activity and exaggerated stress responses, suggesting central nervous system involvement in disease pathogenesis. Environmental factors prove crucial, with episodes frequently associated with stressful events and improvement following environmental enrichment interventions. Urolithiasis accounts for approximately 15 to 20% of cases, with struvite and calcium oxalate representing the most common stone types. Male cats face particular risk of urethral obstruction due to their longer, narrower urethra, creating a life-threatening emergency requiring immediate intervention. Other causes include bacterial urinary tract infections, more common in older cats with concurrent diseases like diabetes or chronic kidney disease and neoplasia, though this remains uncommon. Treatment approaches vary by underlying cause but emphasize increasing water intake to promote dilute urine, reducing environmental stressors, and providing analgesia during symptomatic episodes. The multifactorial nature of feline lower urinary tract disease reflects complex interactions between anatomy, behavior, environment, and physiology unique to feline species. Feline heartworm and cardiopulmonary disease. Heartworm disease in cats, caused by Diarfilaria emytis transmitted through mosquito bites, presents distinct pathophysiology compared to the better-known canine disease. Cats serve as resistant hosts where most inoculated larvae fail to mature into adult worms, with successful infections typically involving only 1 to 3 adults compared to the dozens that may develop in dogs. Despite lower worm burdens, cats experience significant morbidity because even immature worms cause substantial pulmonary inflammation and vascular remodeling, a syndrome termed heartworm-associated respiratory disease. The feline immune response to heartworm antigens proves more robust than canine responses, with intense inflammatory reactions in pulmonary arterioles causing thickening, villus proliferation, and parenchymal inflammation. These changes reduce pulmonary function and may persist long after worms die. Clinical presentations include respiratory signs mimicking feline asthma, acute collapse or sudden death from arterial thromboembolism when adult worms die, and chronic coughing or exercise intolerance. Diagnosis proves challenging because low worm burdens reduce sensitivity of antigen tests designed for canine use, and antibody tests indicate exposure rather than confirming current infection. Treatment options are limited as the adulticide medications used in dogs prove dangerous in cats, and worm death can trigger life-threatening reactions. Prevention through monthly macrocyclic lactone administration provides the most effective management strategy. The geographic range of feline heartworm infection mirrors mosquito distribution, with prevalence paralleling but typically lower than canine prevalence in endemic regions, reflecting the cat's status as an atypical host for this parasite. Japanese bobtail and natural genetic variants. The Japanese bobtail breed showcases a naturally occurring genetic mutation affecting tail development, resulting in short, kinked tails without the vertebral malformations that plague some other bobtailed breeds. The mutation, inherited as an autosomal recessive trait, disrupts normal caudal vertebral development without affecting other skeletal structures, representing a localized developmental alteration rather than a systemic skeletal dysplasia. Each cat's tail shows unique configuration, with vertebrae varying in number from approximately 5 to 8 and displaying variable degrees of curvature and angle, creating tail morphologies described as resembling chrysanthemum blooms or rabbit tails. Genetic studies have localized the responsible mutation, but its exact molecular nature remains incompletely characterized. The breed originated in Japan centuries ago, appearing in traditional artwork from the Edo period and earlier, indicating long historical presence in Japanese cat populations. Unlike the tailless Manx cat, where the mutation causes spina bifida and other serious defects in homozygous form, the Japanese bobtail mutation produces viable, healthy cats without associated health problems, suggesting a fundamentally different genetic mechanism. The mutation demonstrates incomplete penetrance regarding exact phenotype, with significant variation in tail length and configuration between individuals sharing identical genotypes. Cultural significance in Japan elevated these cats to symbols of good fortune, with the maneki neko, or beckoning cat figure, ubiquitous in Japanese commercial settings, typically depicted as a bobtailed cat. The breed's genetics illustrate how mutations causing relatively minor anatomical variations can achieve fixation in isolated populations, particularly when cultural preferences favor the variant phenotype. Feline cognitive decline and aging. Cognitive dysfunction syndrome affects aging cats with increasing prevalence beyond 15 years of age, presenting with disorientation, altered social interactions, changes in sleep-wake cycles, and house soiling. The neurological basis involves age-related brain changes including cortical atrophy, ventricular enlargement, accumulation of beta-amyloid plaques similar to those in Alzheimer's disease, and reduced concentrations of neurotransmitters including dopamine and serotonin. Neuronal loss occurs particularly in hippocampus and frontal cortex, regions essential for memory formation and executive function. Oxidative stress increases with age as mitochondrial function declines and antioxidant systems become less effective, leading to accumulation of cellular damage. Cerebrovascular changes including arteriosclerosis reduce perfusion and contribute to cognitive decline. Clinical manifestations include aimless wandering, staring into space, failure to recognize familiar people or locations, inappropriate vocalization particularly at night, and changes in litter box usage or grooming behavior. Some affected cats show reversed sleep-wake patterns, sleeping during day and remaining active and vocal at night. Diagnosis remains clinical as no definitive tests exist, requiring exclusion of other medical conditions that might produce similar signs including hyperthyroidism, hypertension, and chronic pain. Environmental enrichment, dietary supplementation with antioxidants and omega-3 fatty acids, and medications including selegiline that enhance dopaminergic neurotransmission may provide modest benefits. The similarity between feline and human age-related cognitive decline has positioned cats as natural models for studying Alzheimer's disease and developing therapeutic interventions potentially applicable across species. Feline vision in complete darkness. Despite extraordinary low-light vision capabilities, cats cannot see in absolute darkness as no amount of optical or neural optimization can detect photons where none exist. The tapetum lucidum's light amplification and the RA-dominated retina's exquisite sensitivity extend the lower threshold of functional vision to approximately 1/6 the light level humans require, but this still represents light levels above absolute zero. In complete darkness, cats rely on whisker-mediated spatial navigation, acute hearing that detects prey movement through sound, and olfactory tracking capabilities. The auditory localization accuracy enables cats to strike at sound sources in darkness with reasonable accuracy, while the whisker array provides near-field environmental mapping that prevents collisions and enables obstacle navigation. Some evidence suggests cats may detect infrared radiation through specialized thermoreceptors in facial skin, potentially allowing detection of warm-bodied prey through heat signature, though this capability remains incompletely characterized. The persistent myth of feline vision in complete darkness likely arose from observations of cats navigating confidently in conditions humans perceive as pitch black but that actually contain minimal light from sources like starlight, distant streetlights, or bioluminescent organisms, illumination levels imperceptible to human vision but sufficient for feline rod-based vision. The distinction proves important for understanding both feline sensory ecology and the limitations of even highly specialized sensory systems, as physical laws ultimately constrain biological capabilities regardless of evolutionary optimization. The Flamen response and male-female communication. The Flamen response serves particularly important functions in feline reproductive communication, with male cats performing this behavior frequently when investigating female urine. Female urine contains felinine and related compounds whose concentration and chemical modification patterns convey detailed information about reproductive status, estrous cycle phase, and individual identity. The vomeronasal organ possesses receptors specifically tuned to these pheromonal signals, enabling males to assess female receptivity from chemosensory cues alone. During proestrus and estrus, female cats produce increased quantities of specific urinary proteins that bind and stabilize volatile pheromones, extending their environmental persistence and enhancing signal transmission. When males detect these estrous signals through the vomeronasal system, neural pathways to the hypothalamus trigger gonadotropin-releasing hormone secretion, which in turn stimulates luteinizing hormone release, facilitating the physiological preparations for mating behavior. This represents a clear example of pheromonal control over reproductive physiology, where chemical signals from one individual directly alter hormonal status in another. Female cats also perform Flamen responses, though less frequently than males, using this system to assess male competitive status and possibly to detect predator presence through carnivore urine markers. The ritual nature of feline, with its stereotyped motor pattern, suggests evolutionary refinement where reliability of signal detection justified the development of a specialized behavioral display for positioning scent molecules optimally for vomeronasal organ analysis, emphasizing the critical importance of chemical communication in feline social and reproductive biology. Feline pancreatitis and triaditis syndrome. Pancreatitis in cats. Inflammation of the pancreatic tissue with release of activated digestive enzymes causing autodigestion presents diagnostic and therapeutic challenges distinct from the disease in dogs or humans. Cats commonly develop chronic, smoldering pancreatitis rather than the acute, severe form typical in dogs, with clinical signs including lethargy, decreased appetite, and occasional vomiting remaining frustratingly nonspecific. The anatomic proximity of the pancreas to the liver and intestinal tract in cats facilitates concurrent inflammatory disease affecting all three organs simultaneously, a syndrome termed triaditis involving pancreatitis, cholangiohepatitis, and inflammatory bowel disease. The common bile duct and pancreatic duct join before entering the duodenum in cats, unlike the separate entry points in dogs, potentially allowing bacterial ascending infection or inflammatory mediators to spread between organs. Diagnosis requires combination of clinical signs, elevated pancreatic-specific lipase, and ultrasonographic findings as no single test provides definitive diagnosis. Treatment remains largely supportive, emphasizing nutritional support, pain management, antiemetic therapy, and addressing concurrent diseases. The etiology remains unclear in most cases, though associations exist with preceding dietary indiscretion, hypercalcemia, trauma, infectious agents, and inflammatory bowel disease. Some breeds, including Siamese cats, show increased prevalence, suggesting genetic predisposition. The challenge of recognizing and diagnosing feline pancreatitis stems from its subtle presentation compared to other species, potentially resulting in chronic, unrecognized disease significantly impacting quality of life and complicating management of concurrent conditions like diabetes mellitus, which can both result from and contribute to pancreatic inflammation. Feline hyperthyroidism and thyroid neoplasia. Hyperthyroidism represents the most common endocrinopathy in older cats, with prevalence exceeding 10% in geriatric populations. The condition results from autonomous thyroid hormone production by benign adenomatous hyperplasia affecting one or both thyroid lobes. The etiology remains incompletely understood despite decades of research, though associations exist with consumption of canned food, exposure to polybrominated diphenyl ethers used as flame retardants, and possibly goitrogenic compounds in food or environment. The adenomatous changes cause unregulated thyroxine synthesis and secretion independent of normal pituitary thyroid-stimulating hormone control, creating a state of continuous thyroid hormone excess. Clinical manifestations include weight loss despite increased appetite, hyperactivity, poor hair coat, vomiting, diarrhea, and behavioral changes. Cardiovascular consequences including hypertension, left ventricular hypertrophy, and tachyarrhythmias develop as thyroid hormone excess increases cardiac workload and myocardial oxygen demand. Diagnosis via measurement of total thyroxine concentration in serum proves straightforward in most cases, though early or occult hyperthyroidism requires additional testing including free thyroxine and thyroid suppression tests. Treatment options include daily oral antithyroid medications like methimazole, radioactive iodine therapy providing curative treatment through selective destruction of hyperactive thyroid tissue, surgical thyroidectomy, or limited iodine diets that restrict thyroid hormone synthesis substrate. Methimazole occasionally causes adverse effects including gastrointestinal signs, facial excoriation, and blood dyscrasias necessitating monitoring. The increasing prevalence of feline hyperthyroidism over recent decades suggests environmental or dietary changes have altered disease risk, though specific causative factors remain elusive. The origin of the purr motor pattern. The neural circuitry governing purring behavior originates in the medulla oblongata, with a central pattern generator producing rhythmic signals to laryngeal muscles independent of conscious control or respiratory rhythm. This neural oscillator fires at approximately 25 Hz, triggering rapid twitching of the intrinsic laryngeal muscles that control glottal aperture. The pattern generator receives modulatory input from the limbic system, particularly the amygdala and hypothalamus, explaining why purring occurs in diverse emotional contexts ranging from contentment to severe stress or impending death. The connection to limbic structures suggests purring initially evolved as a social communication signal between mother and offspring, as kittens begin purring during nursing within days of birth, and the vibrations may provide maternal reassurance that offspring are present and feeding successfully in dark nest environments where visual confirmation is impossible. The maintenance of purring into adulthood, particularly directed toward humans, represents neotenic retention of juvenile communication patterns, similar to other features of domestication. Interestingly, most wild felids cannot purr while roaring, with the purring capability associated with ossification of the hyoid apparatus that physically limits laryngeal reconfiguration necessary for roaring vocalizations. Domestic cats and other purring felids possess a completely ossified hyoid, while roaring species including lions and tigers have a flexible hyoid apparatus with cartilaginous elements enabling laryngeal gymnastics that produce roars. Feline asthma and allergic airways disease. Feline asthma affects approximately 1 to 5% of cats, characterized by chronic inflammatory airway disease causing bronchoconstriction, mucus accumulation, and reversible airway obstruction. The pathophysiology involves allergic airway inflammation where inhaled allergens trigger type 1 hypersensitivity reactions mediated by immunoglobulin E antibodies bound to mast cells in respiratory mucosa. Allergen binding causes mast cell degranulation with release of histamine, leukotrienes, and other inflammatory mediators that promote bronchial smooth muscle contraction, mucus hypersecretion, and airway wall inflammation. Eosinophils infiltrate airways in response to these inflammatory signals, releasing cytotoxic proteins that damage epithelium and perpetuate inflammation. Clinical signs include cough, wheezing, increased respiratory effort, and acute respiratory distress during severe exacerbations, with symptoms often worsening seasonally, suggesting environmental allergen triggers. Chest radiographs typically show bronchial wall thickening, creating a characteristic donut or tramline pattern, though significant airway disease can exist with normal radiographic findings. Diagnosis requires ruling out other causes of respiratory signs including heartworm disease, infectious bronchitis, and neoplasia. Treatment employs inhaled corticosteroids delivered through specially designed spacers with masks, providing direct airway drug delivery while minimizing systemic exposure, alongside bronchodilators like albuterol for acute symptom relief. Environmental modifications reducing allergen exposure including air filtration, dust-free litter, and elimination of aerosol products complement medical management. The similarity between feline and human asthma has positioned cats as natural models for understanding allergic airway disease mechanisms and evaluating therapeutic interventions, with findings from feline studies contributing to human medical advances in this common and potentially life-threatening condition. Feline osteochondrodysplasia and cartilage defects. Scottish fold cats, characterized by their distinctive folded ears, carry a dominant mutation causing osteochondrodysplasia, a systemic cartilage and bone development disorder with significant welfare implications. The mutation affects the TRPV4 gene, which encodes a calcium channel protein essential for normal chondrocyte function and cartilage matrix production. The folded ear phenotype results from cartilage abnormalities preventing normal auricular cartilage rigidity, causing the pinna to fold forward. However, the mutation's effects extend far beyond the ears, affecting cartilage throughout the skeleton. Homozygous cats develop severe, crippling skeletal abnormalities evident from young age, including shortened, thickened limbs, fused vertebrae, and progressive arthritis causing chronic pain. Even heterozygous cats, while showing milder phenotypes, develop progressive arthropathy with age, particularly affecting the tail, ankles, and knees. Radiographic changes include shortened distal limbs, irregular epiphyseal surfaces, and periarticular new bone formation. The progressive nature means cats appear normal initially but develop increasingly severe mobility limitations and pain as they mature. Ethical concerns surround continued breeding of Scottish folds, as the desired ear fold cannot be separated from the skeletal disease, They represent manifestations of the same mutation. Some countries have banned or discouraged Scottish fold breeding on welfare grounds. The breed illustrates the tension between aesthetic preferences and animal welfare, where selection for distinctive appearance inadvertently causes chronic suffering through pleiotropic effects of the underlying genetic mutation. Feline odontoclastic resorptive lesions. Tooth resorption, affecting 50 to 70% of cats over 5 years of age, represents the most common dental disease in feline practice. These lesions involve progressive destruction of tooth structure beginning at the cemento-enamel junction, where odontoclasts, cells normally responsible for deciduous tooth resorption, inappropriately attack permanent teeth. The lesions erode through enamel and dentin, eventually reaching the pulp cavity and causing significant pain, though affected cats often show minimal outward signs beyond subtle behavioral changes like preferring soft food or head shyness. The etiology remains incompletely understood despite extensive research, with proposed mechanisms including abnormal vitamin D metabolism, chronic viral infections, dietary factors, and immune-mediated responses. Two types exist based on radiographic appearance. Type 1 lesions show surrounding periodontal disease with maintained tooth and bone radiopacity, while Type 2 lesions demonstrate replacement resorption where tooth structure merges with bone through ankylosis. Advanced lesions may result in crown fracture, leaving root fragments embedded in jaw bone. Diagnosis requires careful oral examination, often under anesthesia, combined with dental radiography to assess extent and type. Treatment involves extraction of affected teeth, though Type 2 lesions with extensive replacement resorption may allow crown amputation, leaving ankylosed roots in situ. The high prevalence and progressive nature make tooth resorption a major feline welfare concern, yet many affected cats remain undiagnosed because dental disease receives insufficient attention in routine veterinary care and cats excel at masking oral pain. Feline parvovirus and panleukopenia. Feline panleukopenia, caused by feline parvovirus closely related to canine parvovirus, represents one of the most significant viral diseases of cats historically, though effective vaccination has dramatically reduced incidence in recent decades. The virus demonstrates remarkable environmental stability, persisting in contaminated environments for over a year and resisting most common disinfectants except bleach solutions. Transmission occurs through contact with infected feces, urine, or fomites, with the virus spreading efficiently in catteries, shelters, and multi-cat households. Following oropharyngeal exposure, the virus targets rapidly dividing cells including intestinal crypt epithelium, bone marrow, and lymphoid tissue. The resulting enteritis causes severe vomiting, hemorrhagic diarrhea, and profound dehydration, while bone marrow suppression produces the characteristic panleukopenia, severe reduction in all white blood cell types, leaving cats immunocompromised and vulnerable to secondary bacterial infections. Kittens infected in utero or during early neonatal period develop cerebellar hypoplasia when virus attacks developing cerebellar neurons, resulting in permanent ataxia, tremors, and incoordination. Mortality rates in unvaccinated, symptomatic cats exceed 90% without aggressive supportive care. Treatment remains supportive, focusing on fluid therapy, nutritional support, antiemetics, and antibiotics for secondary infections, as no specific antiviral therapy exists. Vaccination provides excellent protection, with modified live vaccines inducing strong, long-lasting immunity. The virus's relationship to canine parvovirus illustrates viral evolution and host adaptation, with evidence suggesting feline parvovirus represents the ancestral strain from which canine parvovirus emerged through host switching and subsequent adaptation. Feline social structure and colony dynamics. Contrary to popular perception of cats as strictly solitary, feral and free-roaming cats form complex social structures when resources permit, particularly around concentrated food sources. These colonies typically center on matrilineal groups where related females cooperate in kitten rearing through communal nursing, shared nest sites, and cooperative defense against infanticidal males. Social bonds within these groups involve extensive affiliative behaviors including allogrooming, rubbing, and sleeping in contact, behaviors that maintain group cohesion and distribute colony odor, creating olfactory signatures of group membership. Dominance hierarchies exist but prove more fluid and context-dependent than those in pack-living carnivores, with relationships varying based on resource type and situational factors. Males typically remain more peripheral, though they may associate with colonies, particularly during breeding season. Colony social structure balances the advantages of cooperation, shared vigilance, communal kitten care, collective territory defense, against costs including increased disease transmission, resource competition, and social stress. The ability to form these social groups while maintaining capacity for solitary existence demonstrates feline behavioral flexibility, allowing adaptation to varying ecological conditions. Interestingly, sociability shows individual variation with genetic components, as evidenced by selective breeding producing cats with enhanced social tolerance in some breeds. The domestic cat's social flexibility likely contributed to domestication success, as cats capable of tolerating proximity to humans and other cats could exploit resources in human settlements, while less tolerant individuals remained strictly solitary hunters in marginal habitats beyond human influence. Feline night vision and scotopic sensitivity. The extraordinary scotopic sensitivity enabling feline vision in near darkness emerges from multiple complementary adaptations working synergistically. Beyond the previously discussed rod-dominated retina and tapetum lucidum, cats possess a large cornea and lens relative to eye size, gathering maximum available light and focusing it onto the retina. The pupil dilates to approximately 13 millimeters in darkness, creating a light-gathering aperture approaching the physical limit for the eye's dimensions. The retinal ganglion cells show specialization for detecting motion and contrast rather than fine detail, with receptive field properties optimized for detecting moving edges against backgrounds, precisely the visual task required for detecting prey in dim light. The temporal resolution of feline vision, approximately 50 Hz, exceeds human capabilities, meaning cats perceive visual flicker at rates appearing continuous to humans. This high temporal resolution proves advantageous when tracking rapidly moving prey, allowing discrimination of position across brief time intervals. However, scotopic vision sacrifices spatial acuity, with cats possessing visual acuity approximately 1/10 that of humans under bright light conditions. The absence of a fovea, the retinal region of maximal acuity in primates, reflects evolutionary priorities favoring motion detection and light sensitivity over detail resolution. In conditions where humans see only darkness, cats perceive a world rendered in shades of gray with excellent motion detection, an adaptation enabling the crepuscular hunting strategy that characterizes feline predatory ecology across species from house cats to lions and tigers. The vomeronasal pump mechanism. The vomeronasal organ's function depends on active pumping mechanisms that draw chemical-laden fluids through the narrow ducts connecting the mouth to the sensory epithelium. The organ itself consists of paired tubular structures enclosed in a bony capsule, the vomeronasal cartilage, lying along the nasal septum base. Each tube contains a narrow lumen lined with sensory epithelium on the medial surface and non-sensory epithelium laterally, with a rich blood sinus surrounding the lumen. The pumping mechanism involves the tongue pressing against the palate, creating positive pressure in the oral cavity that forces fluid through the incisive ducts into the vomeronasal lumen. The blood sinus then engorges through autonomic nervous system control, compressing the lumen and expelling fluid back into the nasal cavity, creating a complete pump cycle. This action requires coordination between voluntary behavior, the Flamen response, and autonomic vascular control, illustrating sophisticated neuromuscular integration. The sensory neurons within the vomeronasal organ differ fundamentally from main olfactory neurons, expressing distinct G-protein-coupled receptor families and projecting to separate neural processing centers. These neurons lack cilia, instead possessing microvilli that extend into mucus covering the sensory epithelium. The signal transduction mechanisms also differ, utilizing a phospholipase C pathway rather than the cyclic AMP cascade used in main olfactory transduction. This molecular distinctiveness reflects the separate evolutionary origins of these chemosensory systems and their specialized functions in detecting different classes of chemical signals in feline social and reproductive behavior. Feline coronavirus enteritis. Feline enteric coronavirus represents a ubiquitous infection affecting cat populations worldwide, with seroprevalence reaching 80 to 90% in multi-cat environments. Unlike the mutated, virulent strains causing feline infectious peritonitis, the enteric form typically produces mild, self-limiting gastrointestinal signs or remains clinically inapparent. The virus replicates primarily in intestinal epithelial cells lining the small and large intestines, causing villus atrophy and transient diarrhea in some infected cats. Transmission occurs through the fecal-oral route, with viral shedding beginning within days of infection and potentially continuing for months, though most cats eventually clear the infection or reduce shedding to minimal levels. Kittens face highest risk for clinical disease, particularly following the stress of weaning and maternal antibody waning. The virus demonstrates high mutation rates typical of RNA viruses, creating extensive genetic diversity within and between infected cats. Most mutations prove neutral or deleterious to viral fitness, but occasionally mutations arise that alter viral tropism from intestinal epithelium to macrophages, creating the systemically virulent strains responsible for FIP. The frequency of these mutation events remains low, estimated at less than 10% of infected cats, but represents a constant threat in infected populations. No treatment exists for enteric coronavirus, and management focuses on supportive care during symptomatic periods and reducing environmental stress and crowding that facilitate transmission. Attempts to develop vaccines have largely failed, complicated by the fact that immunity to enteric coronavirus doesn't prevent FIP development and might actually enhance disease through antibody-dependent enhancement mechanisms. The persistence of enteric coronavirus in cat populations reflects high transmissibility, environmental stability, and the predominantly mild clinical course that allows infected cats to remain socially integrated and continue shedding virus. Feline cerebellar hypoplasia from panleukopenia. When feline parvovirus infects pregnant queens or young kittens during the critical period of cerebellar development, it selectively attacks rapidly dividing granule cells in the developing cerebellum, producing cerebellar hypoplasia, a permanent neurological condition characterized by incoordination and tremors. The cerebellum develops postnatally in cats, with the external granule layer generating neurons through rapid cell division during the first weeks of life. This cellular proliferation makes these neurons exquisitely vulnerable to parvovirus, which requires actively dividing cells for replication. The degree of cerebellar damage correlates with the timing and severity of infection, ranging from mild incoordination to profound ataxia preventing independent locomotion. Affected kittens show characteristic signs including intention tremors that worsen with purposeful movement, wide-based stance, dysmetria causing overreaching or underreaching during movement, and frequent falls. The signs typically stabilize rather than progress, as the damage occurs during development but doesn't continue once neurogenesis completes. Many affected cats adapt remarkably well to their disability, learning compensatory strategies and living full lifespans with appropriate environmental modifications. The cerebellum controls motor coordination through complex computations comparing intended movements with actual execution, using error signals to refine ongoing movements and learn motor skills. The reduced cerebellar size in affected cats limits this computational capacity without eliminating it entirely, as surviving neurons can partially compensate through plastic changes. Cerebellar hypoplasia serves as a reminder of the importance of vaccinating breeding females before pregnancy, ensuring maternal antibody transfer that protects kittens during vulnerable developmental periods when their own immune systems remain immature. The Jacobson's organ circuitry to reproductive centers. The neural pathways connecting the vomeronasal organ to reproductive control centers demonstrate the system's function in mediating pheromonal influences on behavior and physiology. Sensory neurons in the vomeronasal epithelium project their axons to the accessory olfactory bulb, a neural structure distinct from and smaller than the main olfactory bulb that processes airborne odors. Within the accessory olfactory bulb, vomeronasal afferents synapse onto mitral cells that in turn project predominantly to the medial amygdala, a brain region critically involved in processing social and reproductive information. From the medial amygdala, projections reach the medial preoptic area and hypothalamus, brain regions controlling reproductive behavior and neuroendocrine function. This anatomical arrangement creates a direct pathway whereby chemical signals detected through the vomeronasal organ can influence reproductive physiology without requiring cortical processing or conscious awareness. Lesion studies demonstrate that damage to the vomeronasal organ or accessory olfactory bulb disrupts male sexual behavior and reduces aggression toward other males, while leaving other behavioral and cognitive functions intact. Female reproductive cycles show alterations when vomeronasal input is interrupted, with lengthened intervals between estrus periods and reduced behavioral receptivity. The system also mediates pregnancy block phenomena, where exposure to unfamiliar male pheromones shortly after mating can terminate pregnancy, presumably an adaptation allowing females to avoid investing in offspring when male turnover suggests unstable social conditions. These effects illustrate pheromones functioning as genuine hormones, chemical messengers that alter physiological states across individuals, rather than simple scent cues requiring cognitive interpretation. The vomeronasal system's privileged access to limbic and hypothalamic circuits makes it uniquely powerful in controlling feline social and reproductive behavior. Feline acromegaly and growth hormone excess. Acromegaly, resulting from chronic growth hormone excess, affects cats with increasing recognition as improved diagnostic capabilities reveal previously undetected cases. The condition typically results from a functional pituitary adenoma secreting growth hormone independent of normal regulatory control. Growth hormone excess promotes insulin resistance through multiple mechanisms including impaired insulin receptor signaling and increased hepatic glucose production, frequently causing diabetes mellitus as the initial clinical presentation. Affected cats demonstrate characteristic physical changes including enlarged head and jaw, increased paw size, interdental spacing from jaw growth, and abdominal organomegaly. However, these changes develop gradually and may go unrecognized until dramatic. Weight gain despite treated diabetes, or insulin requirements exceeding 2 units per kilogram body weight, should trigger suspicion for underlying acromegaly. Diagnosis involves measuring insulin-like growth factor 1, the mediator of growth hormones effects, which becomes elevated in acromegaly. Advanced imaging using CT or MRI typically reveals a pituitary mass. Treatment options include radiation therapy targeting the pituitary tumor, transsphenoidal hypophysectomy surgically removing the adenoma, or long-acting somatostatin analogs that suppress growth hormone secretion and tumor growth. Left untreated, acromegaly causes progressive diabetes, cardiac hypertrophy, hypertension, and eventual heart failure or renal failure. The condition appears more common in older male cats, though both sexes can be affected. Some evidence suggests overrepresentation in certain breeds, possibly indicating genetic predisposition to pituitary adenoma development. Recognition of acromegaly has increased dramatically as awareness grows and IGF-1 testing becomes more available, suggesting the condition has been substantially underdiagnosed historically in diabetic cats with poor glycemic control. Feline lungworm and parasitic pneumonia. Illurostrongylus abstrusis, the feline lungworm, represents an underrecognized respiratory parasite affecting cats worldwide. The complex indirect life cycle requires intermediate hosts including snails and slugs, with parotenic hosts like rodents and birds potentially harboring infective larvae. Cats become infected by consuming intermediate or parotenic hosts, with larvae penetrating the intestinal wall, migrating through mesenteric lymphatics to the venous circulation, and reaching pulmonary capillaries where they penetrate into airways. Adult worms reside in terminal bronchioles and alveolar ducts, producing eggs that hatch into first-stage larvae, which migrate up the respiratory tree, are swallowed, and passed in feces, completing the cycle. Clinical signs vary from absent in light infections to severe dyspnea in heavy burdens, with coughing, tachypnea, and exercise intolerance commonly reported. Radiographic changes include nodular interstitial patterns, alveolar infiltrates, and occasionally pleural effusion. Eosinophilic inflammatory responses to parasite antigens contribute significantly to pathology, with airway inflammation and parenchymal damage potentially persisting after parasite elimination. Diagnosis traditionally required Bearman technique to recover larvae from feces, but larvae shedding occurs intermittently, reducing sensitivity. Bronchoalveolar lavage cytology showing eosinophils and occasional larvae provides more reliable diagnosis. Treatment with fenbendazole, moxidectin, or eprinomectin effectively eliminates adult worms, though inflammatory pathology may require concurrent corticosteroid therapy. The parasite's distribution correlates with intermediate host presence, being more common in areas with high mollusk populations and outdoor access cats. Climate change and increasing outdoor cat populations may be expanding lungworm geographic range and prevalence, making it an emerging concern in feline respiratory medicine. Feline degenerative joint disease and osteoarthritis. Degenerative joint disease affects over 90% of cats over 12 years of age based on radiographic studies, yet remains dramatically underdiagnosed due to cats' remarkable ability to mask pain and the subtlety of clinical signs. Osteoarthritis involves progressive cartilage degradation, subchondral bone remodeling, synovial inflammation, and osteophyte formation, creating painful joints with reduced range of motion. Unlike dogs that develop obvious lameness, affected cats typically show behavioral changes including reduced activity, decreased jumping, elimination outside the litter box due to difficulty accessing it, and reduced grooming leading to unkempt coat. The pathophysiology involves imbalance between anabolic and catabolic processes in articular cartilage, with inflammatory cytokines promoting matrix metalloproteinase production that degrades collagen and proteoglycans. Subchondral bone develops microfractures and sclerosis, while synovial inflammation produces additional inflammatory mediators perpetuating the disease process. The elbows, hips, stifles, and spine represent the most commonly affected sites. Diagnosis requires combining history, physical examination findings of joint thickening, crepitus, reduced range of motion, and pain on manipulation, with radiographic evidence of osteophytes, subchondral sclerosis, and joint space narrowing. Many cats show radiographic changes without overt clinical signs, indicating pain tolerance or behavioral compensation masks substantial underlying disease. Treatment emphasizes multimodal approaches including weight management to reduce joint loading, environmental modifications providing easy access to resources, non-steroidal anti-inflammatory drugs administered cautiously given feline sensitivity to these medications, and adjunctive therapies including omega-3 fatty acids, glucosamine and chondroitin supplementation, and increasingly, monoclonal antibody therapy targeting nerve growth factor to reduce pain signaling. Recognition and treatment of feline osteoarthritis significantly improves quality of life in aging cats. The Persian cat's brachycephalic syndrome. Selective breeding for extreme facial foreshortening in Persian and related breeds has created brachycephalic airway syndrome paralleling that seen in similarly affected dog breeds. The compressed skull morphology crowds soft tissue structures into reduced spaces, creating multiple anatomical abnormalities including stenotic nares with narrowed nostril openings, elongated soft palate extending into the airway, everted laryngeal saccules blocking airflow, and hypoplastic trachea with reduced diameter. These changes increase airway resistance, requiring increased inspiratory effort that creates negative pressure, further collapsing soft tissues into the airway in a self-perpetuating cycle. Affected cats show exercise intolerance, increased respiratory noise including stertor and stridor, open mouth breathing under stress, and increased risk of heat-related illness due to compromised evaporative cooling through respiratory surfaces. The facial structure also creates dental abnormalities including crowding, malocclusion, and increased periodontal disease risk, while shallow orbits cause exophthalmos with increased exposure keratitis risk. Tear drainage issues arise from malformed nasolacrimal ducts, causing chronic epiphora and facial staining. Breed standards emphasizing flat faces have intensified over decades, with extreme examples showing facial structures approaching physical limits of viability. Some breeding organizations have revised standards to encourage more moderate facial types, recognizing welfare concerns. Surgical interventions including widening stenotic nares, shortening elongated soft palates, and resecting everted saccules can improve quality of life but cannot fully correct the underlying skeletal abnormalities. The brachycephalic Persian illustrates ethical tensions in selective breeding where aesthetic preferences have created animals with chronic health challenges affecting daily comfort and lifespan, raising fundamental questions about acceptable limits in modifying animal morphology through selective breeding practices. Feline calicivirus and oral disease. Feline calicivirus represents a highly successful pathogen affecting domestic cat populations worldwide, with numerous circulating strains showing considerable antigenic diversity. The virus replicates primarily in oral and respiratory epithelium, causing acute disease characterized by oral ulceration, sneezing, nasal discharge, and conjunctivitis. The painful oral ulcers typically affect the tongue, particularly the tip and margins, hard palate, and lips, causing hypersalivation and anorexia. Some viral strains show tropism for synovial tissues, causing transient lameness and joint pain, particularly in young kittens following vaccination with modified live vaccines containing these strains. A particularly virulent systemic strain emerged in the early 2000s, termed virulent systemic feline calicivirus, causing severe disease with fever, edema of the face and limbs, cutaneous ulceration, jaundice, and high mortality rates approaching 50%. This strain spreads readily in multi-cat environments, causing devastating outbreaks. The virus demonstrates environmental persistence, remaining infectious on surfaces for weeks, and shows resistance to many disinfectants except bleach. Transmission occurs through direct contact, aerosols, and fomites, with recovered cats potentially shedding virus for months or years as chronic carriers. The extensive antigenic variation among strains complicates vaccination strategies, as vaccines may not provide complete cross-protection against divergent field strains. Treatment remains supportive, focusing on pain management, nutritional support through feeding tubes if needed, and antibiotics for secondary bacterial infections. The virus's ability to persist in populations through chronic carriers and its propensity for antigenic variation make it a continuing challenge in feline medicine, particularly in shelter and cattery environments where crowding facilitates transmission and outbreaks can overwhelm management efforts. Feline herpes virus and chronic ocular disease. Feline herpes virus type 1 causes acute upper respiratory disease but more significantly establishes lifelong latent infections in trigeminal ganglia with potential for periodic reactivation throughout the cat's life. Following initial infection, typically in young kittens, the virus causes severe conjunctivitis, rhinosinusitis, and occasionally keratitis with corneal ulceration. The acute phase resolves over 2 to 3 weeks in immunocompetent cats, but virus persists in latent form within neurons. Stress, immunosuppression, or corticosteroid administration can trigger reactivation with recrudescent clinical signs. Chronic sequelae include dry eye from lacrimal gland damage, chronic rhinosinusitis with nasal discharge and sneezing, and eosinophilic keratitis causing proliferative pink masses on the corneal surface. Some cats develop corneal sequestra, focal areas of brown-black corneal necrosis requiring surgical excision. The virus shows particular tropism for corneal epithelium, and recurrent herpetic keratitis can cause progressive scarring with vision impairment. Neonatal kittens infected from their mother may develop severe systemic disease with high mortality, though this remains uncommon. Diagnosis of active infection uses PCR detecting viral DNA in conjunctival swabs, though latently infected cats may test positive without having active disease. Treatment employs oral antiviral drugs including famciclovir, which achieves good ocular tissue penetration and effectively reduces viral replication during active disease. L-lysine supplementation, theorized to compete with arginine required for viral replication, remains controversial with limited evidence for efficacy. Vaccination reduces disease severity but doesn't prevent infection or latency. The chronic, recurring nature of herpetic disease creates ongoing welfare impacts and veterinary care requirements throughout affected cats' lives, making it one of the most consequential feline infectious diseases despite rarely causing acute mortality in adult cats. The evolution of Felidae and divergence timing. The family Felidae diverged from other carnivore lineages approximately 25 million years ago during the Oligocene, with the earliest feliform carnivores appearing in the fossil record from this period. The family subsequently radiated into two major subfamilies: Pantherini containing the roaring cats including lions, tigers, leopards, and jaguars, and Feline encompassing all other cats including domestic cats, cougars, lynxes, and cheetahs. This split occurred approximately 11 million years ago, making Felidae among the most recently diverged carnivore families. The domestic cat lineage, genus Felis, separated from other feline members approximately 6 to 7 million years ago, with the direct ancestor of domestic cats, Felis silvestris libica, the African wildcat, diverging from European wildcat populations less than 1 million years ago. This recent evolutionary history explains the remarkable genetic similarity between domestic cats and their wild relatives, with domestic cats and African wildcats showing less than 1% genetic divergence. The felid body plan has remained remarkably conservative throughout evolution, with modern species showing strong morphological similarity to ancient ancestors, reflecting optimization for ambush predation that has required minimal modification across diverse environments. Recent molecular phylogenetic studies have revealed 8 distinct lineages within Felidae corresponding to ancient geographic radiations, with domestic cats belonging to the Mediterranean basin lineage. The family's relatively recent origin and rapid radiation have created challenges for phylogenetic reconstruction, as closely related species retain ability to hybridize and insufficient time has elapsed for accumulation of large genetic distances, complicating efforts to resolve exact branching patterns within the feline evolutionary tree using molecular data alone.